Thank you so much, everyone. It's a great pleasure, actually, to be here. This is the first presentation I do after submitting my thesis, so it's a big deal for me, actually. Fun fact, this is my frog. I call my frog because I've been studying them for 5 years. But talking about calls, I don't know if you can hear it calling, but I'm pretty sure it's 2 males, not a couple, so like a amplexus. But I don't know why the second one is not calling. I think it's just pretending or just I don't know, enjoying. But yeah, my name is Sasso Lopes, and so as I said, I'm almost finishing my PhD. I submitted in February, so I'm in the point that I'm replying to the comments from the other scientists that reviewed my thesis before I can call myself a doctor. But I'll present a little bit of my PhD that I did on tracing an amphibian pathogen in the Australian Gondwanan rainforest. So, but before I start, I need to confess that I'm cheating here in the frogs Victoria. My frog and the area I surveyed are not at all in Victoria. They're up north, like 19 hours by the M31, if you want to go see them, in Queensland and New South Wales. But either in Victoria or Queensland, we all know that anywhere in the world, actually, the frogs are not having the great time at all. We have a long list of threats that are actually affecting frogs everywhere. And as we can see, disease is on the top ones based on the number of species that is actually affected worldwide. And no need to say that one of the worst diseases we are studying right now and talking a lot is called Chytridiomycosis, that's caused by this fungus. Oh, you can barely see that. Batrachochytrium dendrobatidis. And in a nutshell, how this fungus grows on adult skins is that it develops this sporangium that is basically a bag full of cells. And when they're ready, this bag opens and releases a lot of motile zoospores. These motile zoospores can reinfect the same frog or can be released in aquatic environment where it can be transmitted and is spread to other frogs. And once you get again in the skin of the frog, it penetrates the skin and develops another sporangium to produce more spores of fungus. So by growing in the skin of the frog, the traditional method that we have to know where the pathogen is out there is by relying on finding the frog, the host, and swabbing its skins to collect any DNA, any spores of the fungus that the frog might be carrying. But because these spores can also survive in the water, it doesn't develop, it doesn't grow or reproduce at all. It just stays in this stage that doesn't develop. But it can stay in the water for up to like 7 weeks. And so another method that we are studying now is to collect the water from where the frogs live to try to find this pathogen, independent of finding the frog, the host itself. This technique is called environmental DNA collection, and it's a non-invasive method because it's just basically going out in the field, collecting water with a water bottle, filtering, and the filtering is just like basically filtering coffee through this paper. And then we collect this paper and track the DNA, extract all the DNA that we had in the water out from this paper that we call eDNA filter. I'm not talking much about the Myxophilis flyi, my frog. This is the star of my PhD, but I can talk about it more later. But this frog declined a lot when the chytrid fungus was introduced to Australia. But now, in the last few years, is showing some signs of recovery in New South Wales. And this frog triggers me to sample eDNA from water to either find the frog DNA and the fungus DNA from where this species is living, because it breeds and it walks and lay eggs in streams in rainforest, but only occurs within national parks and very close forests in this border with Queensland and New South Wales, and there is just a few individuals trying to survive up there, where it used to be a good population. So what I did in my PhD is that I collected water from 31 streams around this area where the Myxophilus flyi could be recovering. And I collected water 4 times. I started in September 2019 in the late spring and ended in May 2020 in late autumn. And in total, at the end, I collected 230 filters of DNA, and there was nearly 460 liters of water that I was filtering. I start filtering in the field, but I don't know if you try to, it seems clean, but when you filter, even if you think the water is super clean, the filters get like very dark brown, so it's very hard to pump in the filter. So I decided to change my methods and took it to the lab to use the machine to pump, because otherwise I couldn't finish that amount of water. So what I've seen after all those water filters is that all across the area where I surveyed, the DNA detection varied a lot. So each of these circles shows how much of the eDNA of the fungus are found in the water, and also how many of the replicates that did it in the stream were positive. So imagine I collected water at 5 water bottles, and how many of the 5 water bottles from that stream were actually showing the fungus. So here, so the size is how positive I was that I found the BD in the water, and the color is how much of the BD I found it. And as you can see, it varied a lot between streams across Queensland and New South Wales. But also these ones that are showing darker and bigger graphs are actually sites where the Myxophilus flyi is doing great. So it's very ironic that where I found it more BD in the water, the frogs, they are the favorite sites where we actually see, and the frogs, they're supposed to be the best sites for flyi. But when I combine all the streams together and see if there is any pattern across the seasons that I survey, I see in that graph, for example, it says how much I found it. So how many replicates had the fungus. We see that only in the first season was different from the others. It was the highest. So this was around September that I collected water. It has the highest positive BD in the water. And when I see how much of the eDNA of the BD was in the water, there was no significant difference. But you can see that in the second season, this was around December 2019, we had a slightly more of the fungus in the water than any other season. And I don't know the reason it could be from that, but I don't know where the graphics show. Yeah. But if you compare to the frogs, the fungus that we found in frogs, it doesn't relate it to the frogs. So I swabbed the Myxophilus flyi everywhere on those streams, and as you can see, and only in the season 3, the frogs had a lot of the fungus, and not in the second season, as we saw in the water. It's not showing here, sorry, but I had another graph here showing how the temperature and the rainfall was changing across each season. And I don't know if you remember, I hope so, because it was very drastic in Queensland, in New South Wales, and also here in Victoria. In December 2019, when I found that more of the DNA copies in the water, was also the time that we had the worst bushfires happening in December. So I remember having to drive to the field and actually have to turn around and go back home because the national park was being closed to evacuate because it was starting to get fire all around the park. So it was very dry and very hot. So one of the explanations that we came up for finding a lot of BD in the water was actually those streams stopped flowing at that time because there was no rainfall at all, and formed these ponds where BD was just forming this pool of fungus and that was accumulated, and that's where I collect water. In contrast to when there's a lot of rainfall and the stream flow a lot of water, so it just washed away everything, including the BD. So at that time, when there was a lot of rainfall, I couldn't find the fungus in the water. So if you ask me if you should use eDNA to find BD in the water for your work or for saving frogs, I'll say yes, definitely. It has some issues, but also provides an image of the exposure of the fungus, the amphibians, to the pathogen. You can collect it any time in the day or many times. It's faster than trying to find the frog, unfortunately. So you can see if there is the BD more often and in a larger scales, because you can just go everywhere collecting water. But we have to remind that the presence of the BD in the water is not, as we saw with my frogs, it cannot, sometimes it's a balance between the frog input, so it actually cannot reflect how much of the BD we find in the frog, but it's also a matter of the environmental conditions. So if it's raining a lot, you'll be just wash the DNA away, you'll not find the pathogen there. And you might think that the BD is gone forever and everything clean, but actually it's just because the rain washed it away. So you have to take these things into consideration if you want to use environmental DNA to detect the pathogen. And in one of my thesis, I think one of my comments on my thesis was like, what is the matter then? What is the point of using eDNA if you can just find the frog? You just find the frog, you know where the fungus is, and problem solved. And then I have to convince why sampling water or sampling environment is important. And it's, well, although we like to think that when we go to find frogs, we are great in finding frogs, we actually miss them a lot. It's very easy for us, for humans, to miss frogs. And by sampling water, if you only depend on finding the frog to swab it and know if the BD is there, you might be missing data there because you don't find the frog, as in this image. I don't know, maybe you can see, I don't know if you want me to tell where the frog is. Or, yeah. So it's this. So these are the legs, and this is the head, and this is one of the arms. Can you see it from? Yeah, so that's why environmental DNA. We also applied selecting soil to try to find either the frog DNA or the fungus DNA also from soil. And I think that's the great from sampling environment, that you can just help yourself to when you can't find the frog. And that's all. Thank you so much.